I think that human beings are visual animals. We gain a lot of information just from shape. In some cases, a structure is really very meaningful. Structure information is just amazing.
So, how can simply observing something bring on a breakthrough? Let's find out. Listen as Eva Nogales describes the power of observation and the idea that sometimes the most straightforward approach can be the most revealing. Eva's story reveals the benefits of staying motivated in the face of failure. It's a testament to the importance of close and purposeful observation, and proves that critical thinking and persistence can give rise to groundbreaking discoveries. You found Heureka. First, just for the record, could you state your name and your affiliations?
Yeah, Eva Nogales, and I'm a professor of molecular cell biology at UC Berkeley, a Howard Hughes investigator, and a senior faculty scientist at the Lawrence Berkeley National Lab.
That's perfect. You got that one. Okay. Eva, who was born in Spain, is a professor of molecular biology at the University of California at Berkeley and a senior researcher at the Lawrence Berkeley National Labs. Visualization is crucial to her research methods, which revolve around using electron microscopy and computational image analysis to better understand macromolecular complexes. This methodology has brought about major discoveries with regard to the treatment of diseases like cancer and garnered critical acclaim for Eva and her lab. If you were going to come across a brand new gene or protein and you know nothing about it, what's the first experiment you would do to learn something?
This is not the kind of thing that happens in my lab, because we always tend to pursue something that it has been already characterized in terms of identifying what it is and what it does, and we want to learn the how it does it. But I presume this day and age, I would maybe find the right cell type and knock it out and see what happens, for example, or try to tag it and pull it and see what it comes up with. But just realize that's not normally what we do. We come towards the end of the story of a biological system. We already know what it does, and we already know how relevant it is.
So then why don't you build on that? So tell us a little bit in some lay terms, what is it that you and your lab like to do?
Yeah, so what we like to do is gain a mechanistic understanding of macromolecular function through visualization of the structure of the macromolecules that carry out a certain cellular role that we know is relevant. The concept is very primary. I think that human beings are visual animals. We gain a lot of information just from shape, from motion, and this is the kind of thing that we try to see. When it comes to macromolecules, it actually depends. In some cases, a structure is really very meaningful. Just think of the DNA structure, which is the ultimate prototype, is what launched structural biology as a field, where visualizing the structure tells you so much about how DNA stores information, how it can be repaired, how it can be copied. Then came the structure of hemoglobin, and maybe in that case, it was a little bit less informative what it is that the molecule did just by looking at arrangement of alpha helices. But in many cases, it does tell you a lot. And in cases where the structure of the molecule is mechanical in nature, so it has pieces that use energy to move and push and pull, the structure information is just amazing to give us a conceptual understanding. And ultimately, I think we want to put this in the context that actually are attractive to both scientists and non-scientist citizens, which is, can we put the genetic mutations in the context of our structure so we know what it is?
Yeah, and you're talking about the power of observation. Mm-hmm. And it's interesting because you're describing this deep, fundamental human nature desire to see and to look at and to understand something. And I would absolutely agree that there's something both beautiful and important about being able to see the thing that you want to study.
Mm-hmm. That's right. That's right. I think the concept of structure being a framework where everything gets integrated and made sense. So sometimes we came out, our structures give rise to hypotheses. In many cases, the first step is that the structure integrates a lot of biochemical data that is more abstract, like you say, but in a way that now it does make sense to us in physical terms. So it is that framework for integrating information and hopefully then being able to both interpolate that information in the context of the structure and extrapolate to new knowledge. But that concept of framework that help us think about the process, I think is very powerful, at least for me.
So then go ahead and tell us one of the experiences you've had and some of the challenges and struggles with solving structures and really trying to visualize what proteins look like.
Yeah, so I thought I will tell you an example where I really think with one of my students, I live one of these Heureka moments that you like to recount. And just to give you a little bit of background, this came after a number of years working on the field of tubulin and microtubules, where as a graduate student, I was fortunate enough to be exposed to the technique of cryo-electron microscopy at a time when it was really just starting up the first cryogenic holders.
Here,
for the samples.
Eva is describing a technique called cryo-electron microscopy. Cryo-electron microscopy, or cryo-EM, describes the process of freezing down a sample very, very quickly. And then with that frozen sample, you can use a special type of microscopy to look at very, very small structures. Eva, very early on, was studying a protein called tubulin that instead of forming into long tubes, formed a two-dimensional sheet that was ideal to work out some of the field's earliest technical challenges in cryo-EM.
Just hit the right method. I got a burst of images and I was able to get the structure. And very quickly I went from being in a lab that no one knew to we suddenly were center stage, and I got invited to everybody, to every conference on cytoskeleton. So it was really fantastic. And then I landed at this position at UC Berkeley. And at that point, I had the opportunity to branch out and get into a field that was completely new. It was at the point, it was exciting because I didn't know what was coming. I should have been really scared, but I was just excited. And even in a disappointed way, in the first year, things went very well. We published the first structure super quickly at low resolution, but this was transcription factored over a megadalton complex that contained the Tata binding protein that bind to Tata boxes.
When Eva first started her lab, she was working on a family of proteins that contained something called a Tata box. The TATA refers to the part of the protein that is able to bind to TATA sequences on DNA. One of Eva's main challenges was to visualize how proteins that contain these domains bind to DNA. See, getting a structure of the protein by itself was easy, but getting a structure with it bound to DNA proved much more difficult.
And then for years, we struggled. We were able to get a structure, but we were really interested in how the complex bind DNA, because again, the whole point is to see these molecules in action. And this large macromolecule has as a function to bind, recognizing very specific sequences of DNA. Something happened because we got the APO structure, the one without DNA. Then we started taking images, and we could see in the single individual images, we could see this linear density that was the DNA. We were so excited. Oh my God, we got it. The complex is bound to DNA. We had actually optimized the DNA. So we start with these individual images where we see the DNA. We use our method, which use the reference, the APO complex, to now align the images. And every time, we end up with these images with DNA, and we end up with a structure without DNA. It was desperate. It was like a nightmare. It didn't even make any sense. And we thought we are so close to be able to visualize this, but something gets lost in the pipeline. So this complex, this is getting into the details, but this complex is very large, but it's basically three main lobes, which are make like a horseshoe shape with an opening.
Here, Eva is describing a Eureka moment, where while attending a conference, she and her student realized that one of the questions that had bothered them for so long, how a protein could move such great distances, must be flipping from one arm of a horseshoe to the other side of the horseshoe. This was previously unprecedented for a protein to be able to make such a large jump, but they thought this must be the solution to their problem.
Across. So this is lobe A, B, and C, and lobe A sometimes is attached to lobe B, and sometimes it's attached to lobe C. This is a movement of 150 angstrom. This is a movement that is much bigger than the size of is as big as the size of the lobe. And what is happening is that we were, without the DNA, we have this structure, we're in one state. And once we bind to the DNA, the lobe has moved completely across in order to bind the DNA. And just realizing there is this motion, and it's only one state that bind the DNA is like, oh my God, this complex is being able to control whether it binds or not bind DNA through these gymnastics that mean a completely reorganization. So this was one of these Eureka moments where the struggle, solving the struggle actually meant we now have a true biological story. That was so very exciting.
So then what happened next? So that key insight leads you to this realization that you've got multiple states that can be represented in this projection. So how then do you take that forward to develop on that and to really get the deep insight that you were striving for?
So the most important thing was that this really annoying property of the complex was actually meaningful, that it actually had a function, and that the function had to do with binding the DNA. But at the time, it was really complicated, deal with all of this conformational variability. So what we did is, now with that insight, we said we have to develop a method to simplify the problem so that we can get a grasp on at least one of the structures, and that will be an anchor point to understand the rest of the states and what motions occur.
Understand the complexity of these states. So if you have then these three legs and one of them can essentially pivot between this left-hand side or right-hand side, then when you freeze the solution with the protein in it, that then you subject to the cryo-EM, do you then either get it in the right-hand state or the left-hand state, or do you also capture the intermediate states in between?
Yeah, so the intermediate states are there because this is not teleportation where instantly one lobe move 150 angstrom from one to the other.
Yeah.
But it is true that the intermediate states are relatively short-lived in the sense that in one side or the other, there's a contact that gets established. And then there's a certain off rates and the thing transitions. Exactly. And the two end states are more unique, while the ones in the middle are more random. There's a tether and there's a certain path, but they are less defined. So at the end, if you want, the complex stays longer in those two end states than at any one given state in between.
But you have a pretty complicated mess of all.
We had a really pretty complicated mess. So this is very complicated. We're not overexpressing it in a heterologous system in insect cell or bacteria or anything like that. We were purifying this from HeLa cells using immunopurification, so using an antibody and then eluting from the colon with a peptide. The quantities of TF2D in cells is very low. So we're getting minute amounts. We have to purification, we check with silver stain or something that had that kind of sensitivity. So we cannot do what crystallographers or structural biologists, cryo-EM people are doing when they work with something that they produce in large amounts, either because they purify it from the cell in large amounts like ribosomes, or because they are smaller and they're able to more easily express them. We have so very little, we cannot just bind DNA and then do a size exclusion colon or one of these traditional methods. We have to work with extremely small volumes at a time. So we had come on in the lab with a method to use coated magnetic beads where we could add the DNA and then add the factors that we want to bind to the DNA in excess allowed to incubate them. And we're talking about volumes of 10 microliters where the concentration of our proteins are in the tens of nanomolar, okay? And we incubate, and then what we're able to do is wash the beads for just to get rid of anything that is not bound, relatively stably bound to the beads, and then elute the DNA. We use a restriction enzyme, so it just cuts the DNA and is released from the beads. And then these, we use the magnet, separate the beads, take the sample, put them on an EM grid, and maybe we have enough to make 1 or 2 grids. I just hope for the best, okay? But in any case, really difficult. Even before we start concentrating on the conformational variability, this is very difficult from the biochemical point of view because of the rarity of the complex. But in any case, once now we could concentrate on this structure bound to the DNA, really push the resolution. This was now entering the time where the cryo-EM field exploded with new detectors that gave us better images and things like that. We were able to see that in that stably bound state, the lobe that moved had split into 2. One that we could identify as being TBP that was bound to the DNA, and then the rest was that lobe now without the TBP flopping around because it had done his role. What we could see in the structure is how the complex itself binds at one end and then serves as a ruler to position the factors and the surfaces that are going to be able to engage TBP in the right position to be able to bind the DNA. And the complex is holding the DNA like with 2 hands, with the DNA that is between the 2 hands corresponding to the transcription start site where the polymerase is going to be loading. It's just amazing. It's like an offering, okay? So after the TBP has been placed at the right place, only after release because of recognition of the DNA on one hand, now TBP is positioned to recruit the factors that will put the polymerase between the 2 hands. It was amazing. Honestly, it was really exciting. The details came from then going back to now that we have this structure, let's go back to the mess. Let's go back to a situation where we mix all the factors and we define this conformational landscape, and we see how things move from one state that is completely inhibited. So to me, this took, by the way, it took 20 years. It took 3 consecutive graduate students, each one of them making one more conceptual leap. So yeah, these molecules are doing these processes, but they're always regulated. They're always turned on or off. They're pushed in one direction or the other. And imagine if something is very rigid, how do you modify how it works? If it has many moving pieces, you can really come something that push them in one direction or the other, that jam one elbow but facilitate the movement of the wrist or something like that. So this is, I think, a beautiful, huge complex that is able to integrate signals from the chromatin, from other cofactors, gene-specific transcription factors, in something that ultimately has as an output the recognition of the core promoter and the loading of the polymerase. I think is very cool. I'm not saying that we understand everything. There's a lot of still opening questions that are going to keep us interested and busy for many years. But this is, I think, a conceptual framework that can guide a lot of people, not only us, to do further work and put it in that context.
So then if you give us a sense, specifically of the conceptual framework, so the conceptual framework that you can now apply to other types of problems.
Yeah, so I think this is what a technique that does not require a crystallization process, breaking down into pieces or stabilizing a single state can do for you. You can vary the conditions that are relevant and important for the function, adding factors, adding regulators, changing the sequences of the DNA, and see how at every point the landscape changes, the structural landscape, how different states are occupied and used. It tells you about how it can change in the progression of a certain activity, where different molecular players can be added or removed from the scene to control the process.
So give us a sense then, how often in cryo-EM do you end up with what we might call artifactual structures that are absolutely present in your cryo-EM image, but that might not or were later discovered to be irrelevant for some biological system or biological state? How often does it happen?
I don't know that it has happened yet, that there's been a structure that then has turned out not to be relevant. But what it is true is that there may be some structures that are there that we end up not describing. And this is because at the end of the day, in the cryo-EM process, where you have many different images from many different angles that would correspond to different states, you put some of those images together into a structure based on consistency principles. Okay, so what you group together are images from different views that end up really giving you a three-dimensional structure that is self-consistent with all the data that has gone into it. Now, if states are less represented, maybe it's a lot harder to fish out the few images that correspond to that state to give you the structure. And so less represented state, you may miss. You may not capture them. It doesn't mean that the ones that you obtain are not meaningful. It just means that it's not the full story. But again, think of this in the context of how we were able to do this kind of a study before something like cryo-EM was powerful enough to be able to computationally separate different states.
So one of the themes that I think is going to emerge is a theme about observing and the power of just looking and seeing. And I think this is something that you might have a unique perspective on, given that you've based your entire career around the power of seeing things.
What I thought is that I decided to branch out into something that was completely unknown, that to start with at the beginning, it seemed like it was going to be very easy, and then became a nightmare kind of problem where we could not understand. There was like some logic flaw that didn't make any sense. And we just struggle until there was a total Heureka moment where I said, oh my God, it all makes sense and it's so cool. But it was funny, like you struggle with, in a certain field, you make a breakthrough, and instead of just keeping cozy, you branch into something else and you struggle again. And it's always the struggle.
So the reason I ask is because if we think about the power of observing, high fidelity and confidence in the thing we're observing is going to be really important. So the technology has rapidly developed, in my mind, at an extraordinary pace in order to allow us to see, most recently, to see molecules. The small resolved benzene rings and drug molecules is really remarkable. But of course, the next evolution in all of this are some of these algorithms that are computationally predicting structures.
Yep.
And the major question, of course, is how much fidelity do those computationally derived structures have to the real thing? So I want to ask you what your opinion is on this wild computational future of structure prediction, and how you see that fitting into the sort of tried and true cryo-EM and other structural biology approaches.
Yeah, so when the AlphaFold published the results, and everybody got really super excited, but I also, for a couple of weeks, every seminar or every presentation that I made, that I was giving, people would ask me at the end, so now with this protein structure prediction, are you guys going to experimentally be out of work? And I told them, not, quite the opposite. This is all very welcome. So right now we're at a stage where, in order for us to know that they are successful, you want to do the experiment and see that it coincides. I think is gaining, is gaining momentum, and that we are at a point where we will be prepared to accept as a first approximation that the structures that can come out with are going to be correct. Again, this will be it will take a long time. Definitely, I'll be retired, so it will be someone else's problem at that point. To go from these structures that tend to be single domain, fairly small, being predicted reasonably accurately, to work on multi-subunit complexes where you are looking at proteins that fold in some cases on each other.
Eva is describing here how the need to go from a computational prediction to experimental validation will take time. And really, she's saying that this will first start with some of the easiest structures to validate, but some of the more difficult structures to validate, such as proteins that have multiple units that are placed together in intricate puzzles, will take much, much longer. In fact, much longer than she will still be performing cryo-EM.
Of course, as more and more information is generated experimentally, I think probably these predictors are going to get better and better as well. But I find it, exciting and synergistic right now with many of the type of things that experimentalist structural biologists are doing right now.
So instead of replacing your jobs, it might just, allow the students, instead of 20 years and 3 students, it might reduce the amount of time it'll take a while, hopefully. So then just to begin to wrap up, I'm curious then, when a young student or fellow comes to your lab, what are some of the most important things that you would want to impress upon them?
Yeah, so I think this is in general from anybody that works in science. To me, the most important thing is critical thinking. I just cannot stress it enough. Critical thinking is the sophisticated, technical, scientific way of saying common sense. Is constantly playing devil's advocate for your own data and your own conceptions. And this is not trivial because, I find it easier to play devil's advocate from someone else's data and ideas. And sometimes I get stuck on my own preconceptions or models that I really like because I find it maybe aesthetically pleasing, but just critical thinking. And it is a quality that I really think it is so needed for everybody, even non-scientists. If we were able to be critical thinkers, we would not be taken for a ride by politicians, for example. And we, as a society, as a planet, we will be doing a lot better. But that is the most important thing. Sometimes we just get so excited that we may be just carried out by emotions without being absolutely critical in our thinking. And the analysis of our data, which then allows us to think of better experiments, better ways of doing it, better ways of interpreting the data that we have. So critical thinking is one. Persistence in the face of apparent failure. I always tell people, if we are doing things and we are not failing, is because we're doing things that are trivial and that someone else has done and is all sorted. There has to be a certain amount of failure to move forward. It's just a question of whether failure defeats us or failure informs us, and to whether we learn or not from that failure. Failure in itself is not good, but it doesn't have to be bad either. And if we are working as scientists trying to push the frontiers of knowledge, and with the limited technology that we have at any given point, there is bound to be failure, and people do not have to take it too personally, meaning they don't have to think it is because I'm useless, because it is me that is not able to do it, because that leads to depression and it doesn't take you anywhere. But just be aware, we all struggle. I think sometimes people that appear to be very successful should talk more about their failures to students, because the students will realize it's not only me, they also struggle. This is part of our job. We just need to use that failure to take the next step in the most informed way, and to keep track of what we are doing using common sense so that we move forward. And that resilience in the face of failure, learning from, I wouldn't call them mistakes, but the difficulties of the project itself, they're very important. And I have to keep reminding myself because I'm human. And the other thing is, once you do succeed, rejoice and celebrate the successes, because that will energize you for the next challenge and the next bottleneck. And sometimes, again, we forget to celebrate when things go well, and that is a necessary step. So those just a few things.
Yeah, I want to thank you for sharing your successes and your failures and your resilience, and really pushing the envelope to try to see better and to see things we haven't seen before. So thank you for sharing your story with us.
Thank you for having me.
Thanks for joining us to hear Eva's story. Her discoveries in the field of structural biology reveal the power of observation. Eva reminds us that when we get stuck, we should go back to the basics, keep it simple, and be deliberate. And once we find success, we should always celebrate it. If you liked this story, give us a review on Apple or wherever you listen to your podcasts, and give more people the opportunity to learn from the world's best scientists. Next time, we'll hear a story from Cynthia Kenyon, a pioneer in the study of aging, who shares her story on the importance of curiosity. Want to be featured on our podcast? Luck and serendipity are important aspects of scientific breakthroughs. If you're a scientist with a story about luck to tell, reach out to luck@eurekalabs.org, and you may just end up on our show. Special thanks to the Eureka Stories team. Our sound editing is done by Anna Marquis. Our copywriter is Nicole Kagan, and our media and website team members are Benjamin Osamani and Matthew Norman. To stay up to date, you can follow us on Twitter or Instagram @Eureka Labs. You can also find show notes, transcripts, and video on our website, stories.eurekalabs.org. That's stories.eurekalabs.org. You found Eureka.